thanks for coming, and thanks, Dag, for the invitation. It's really a pleasure to be back in Lund. I actually lived here for 6 months when I did my master's, so it was a long time ago. And I'm happy to be back and give a small talk about large genome assembly. I'll skip the introduction. We can read what's here. The slides will be online, I think, as well as the video, so all of that is text and how to reach me and so on. I mainly did contributions in bioinformatics from a computational point of view because I'm actually a computer scientist. That's a confession that I make here to you here. But I moved to bioinformatics to do more applied stuff to biology. So I did typically two things. One is develop software and methods, and the other one is to do collaborations with biology groups. And I just reminded myself not to point too much because it's an ambiguity on where to look for you. If I point to this one, then you don't look to these ones anymore. So I will not point too much. What before that? Jak prata IT svenska längre? Sorry. I had learned a long time ago, but I don't speak it anymore. This talk is about large genome assembly, and I thought about putting large eukaryotes because it's really about large eukaryotes. But if I put the title this way, there is an ambiguity because you would think is it either large genomes or large organisms? And it's actually going to be large genomes. It fits very well with big talks, I think, for the topic. But for those of you who came thinking it could have been large eukaryotes, I don't want you to leave disappointed, so this is for you. And actually, that's the largest eukaryotes, blue whale, 30 meters. And the content of this talk is going to be shortly talking about genome assembly, shortly mentioning software methods, and the bulk of it will be about giraffe, spruce, and axolotl genome, some experience I've had assembling those genomes, and a few considerations about present and future sequencing technologies. So if you're OK with this, let's proceed. This is a slide that some of you have seen at Chesky, Krumlov workshop, which is just my definition of what assembly is. So if you're not familiar with the process, I assume many of you are already. So you have a genome consisting of many chromosomes which are sequenced into reads that cover it. And then an assembly is what you think the genome is going to be. It's an approximation. So it's a set of sequences which best approximate what you have sequenced. That's the assembly for the genome case. Another slide that some of you might also have seen is what do you do with an assembly? What are the example uses? So of course it's to generate a reference. And notice that I'm actually quoting somebody who knows what to do with an assembly, not myself. So Mike Zodi is an expert in this. So you can generate a reference genome, but it's also used for finding variations between genomes if you don't have a good reference to use. And finally, a very current topic nowadays is to find structural variants. Assembly helps a lot in this direction. Some prehistory of genome assembly. So I will not talk too much about it. That's only one slide. In the Sanger era, when you had only one type of sequencer, it was a good time. There were greedy algorithms for assembly, which basically boils down to finding a way to align your reads one by one and find an organization of them. But it wasn't a very principled way of doing assembly. It worked. But in my opinion, the era ended in 2001 and 2005 when more advanced techniques for genome assembly were proposed. And I'm not going to quiz you by asking what happens in 2001 and 2005. I've did it in some audiences, and not so many people actually know. They typically answer human genome sequencing happened. But actually, in my opinion, something else happened in 2001 and 2005. The publication of those two papers. The first one from Bevner and colleagues introduced the Bruin way of doing assembly. It was previously proposed by other authors, but I would argue that this paper is really the application for modern assembly. And the second one is about string graph formalism, which is the way we assemble genomes using PacBio and nanopore nowadays. Notice this relatively small number of citations for these two papers compared to the broad application that has been used from this proposal. So very important papers, low citation count, and still very important contributions. So nowadays, assembly is performed using two types of pipelines, depending on what type of reads you have. So it's very technology dependent. If you have short reads, then there is a pipeline using the Bruin graphs, removal of error variance, repeat resolution, and in the end, you have an assembly. If you have long reads, it's a completely different type of pipeline that starts by sometimes correcting the reads and then doing a different type of graph and doing some other stuff, and finally you have an assembly. What's going to happen also is it's possible to merge those two types of pipelines and get an hybrid assembly if you have two types of data sets, and there are some benefits to doing this. Although it's not clear nowadays if this type of hybrid assembly, pointing to all three for consistency, a hybrid assembly is better than a long read-only assembly. Maybe the future will tell us which one of the two is better. I would tend towards doing long read only, but hybrid still has some advantages in terms of polishing. I used to work on the short read era because there were only short reads, and nowadays I'm also working in what comes after assembling for long reads. I'll quickly mention it at the end of this talk. So the first part of this talk will focus on things related to short read assembly, and then we'll move on to long reads in roughly, I don't know, 20 minutes or something. And by the way, if you have a pressing question, I know this is embarrassing to be on YouTube for you, but you can always ask it. I'll take questions online if you feel like it. But there will also be times for questions at the end. This is going to be roughly the only technical slide about what's going on in an assembly pipeline. It's very simplified. So it starts from the reads. Imagine you have 3 reads. Then reads are cut into smaller parts that we call kemers. And we find overlaps between them, and we build a graph out of it. Then there is a critical step, which I call refine, so refinement, which cleans a little bit the graph, removes errors, removes other stuff. And finally, after these steps, contigs are created. So from 10,000 feet perspective, that's how short reads are assembled. I'll quickly zoom in a little bit into step 2, because I think it's a critical step. So the graph is constructed. It's typically a Debugging graph, and it contains 2 types of events. The red events correspond to sequencing errors, and the blue events correspond to variants, like SNPs, small indels, inexact repetitions. And those blue events, as well as the red events, are removed from the graph before contigs are constructed. This has 2 consequences. The first is that typically assemblers do a good job at ignoring sequencing errors, and in the assembly you don't get sequencing errors in short reads. The blue events are removed, and that has a significant consequence in genome, transcriptome, and metagenome assembly, which is that you don't see SNPs anymore. If you are assembling a heterozygous organism, deployed or highly heterozygous, deployed, or a metagenome with many strains variations, the job of the assembler is not to report everything, but to report a consensus. So it removes one of the 2 blue paths, and it only keeps one of the 2. So that's what you get in the end in the contigs. In some cases, you might get, like if you see this one longer path, you might get some strain variations, some heterozygous, some different alleles, but not always. Something to keep in mind. And then there's a few steps coming after. The contig step is scaffolding, long-range improvement, and annotation. I'll quickly mention it in the next slides. Custom analysis would be anything you do after annotation. So analyzing genes, and so on, and repeat elements, and so on. I'll go quickly on this one just to make you refer to this slide. So what scaffolding is to improve an assembly by taking into account paired-end information. It's important for having a higher contiguity, longer sequences. It's also a clear source of misassemblies. So it's not clear if you should do it or not, depending on the application. And it's also slowly being made obsolete by long reads. What I would find more interesting nowadays is all these types of technology for long-range improvement of assemblies. So I've listed only 3 of them. One is bio-nano optical mapping techniques. Other one is high-C and dovetails, which are somewhat similar in spirit. And finally, the third one would be linked reads, which is known also as the next genomics for a company that does it. And all 3 techniques really can be used for more than just assemblies, such as finding structural variants and so on, but they can also be used to improve an assembly. So typically, in a typical sequencing project where the aim is to get a good genome, then it's recommended to do one of those 3, if not a combination of those 3. I'll quickly mention some results on, I think it's going to be bio-nano later. And in my opinion, all 3 are valid ways to increase contiguities of assemblies. Just one slide on the evaluation of assembly, and just quickly to mention that there are some metrics that can be computed to see how good an assembly is, and some are very straightforward, such as the total size and so on. And 2 tools are very useful for doing this. One is QuAS, which computes all the metrics. The other one is Bandage, which visualizes the assembly graph. Those of you who have been to workshop on genomics have already seen me struggling to explain the N50 to students, because it's really a mouthful to pronounce this whole sentence. And you will notice that I've slightly improved the slide for next year, so that now there's a more intuitive explanation of what N50 is. It corresponds to just, you take the contigs, you sort them, and you take them up until 50% of the assembly, and that's your N50 size of 1 here, 1 megabases. So there's a way to compute it, and it basically is not so complicated. That number pops up a lot in publications. I'll just flash this slide concerning recommendation of whatever software for assembly you can use depending on the data site, on the data type. I'll move on to giraffe genome assembly. So I was involved in sequencing the giraffe and okapi some years ago. And back then it was 2012 style sequencing technology, so Illumina, with short reads and paired end. I'll argue nowadays it's still a cheap way to get a genome sequenced, but doing only short reads would be kind of strange in the presence of long reads. Maybe it could be motivated by many reasons. So back then we had 30x of paired end and 8x of one of those fancy mate pairs from Nextera, which covered between 4 and 8 kb span. It was very on the low end of sufficient. When they came to me with this data, I said, really? That's all you have? Yes, that's all we have. So no, please assemble it. OK, so we had to do it. So I used the normal strategy back then, an assembler, which was actually not mine, but it perfectly worked fine. It gave us this type of N50s. So for those of you who are familiar with it, it's a reasonable assembly, but nothing spectacular. So good enough contigs to do the analysis, on average 50 kb contigs, and scaffolds are a little bit longer. I would say mediocre would be the correct adjective for this assembly. It was OK, but nothing crazy. What came after is a little bit more interesting. So I delivered this assembly to a biologist, and then they were able to do something with it. So they did some analysis on genes only, and did a comparison between the assembled giraffe and the assembled okapi, which was assembled using the same type of sequencing. And in the end, using those 2 assemblies and also the cow genome and some annotations for the cow, they were able to find 70 genes that showed multiple signs of adaptation related to the long neck of the giraffe. So those genes are putatively the ones which make the giraffe having a long neck, because the giraffe has it, and the okapi, which is basically a giraffe with a short neck, doesn't have them. So that was a quote from the paper. So the stature and cardiovascular adaptation for the giraffe evolved in a small number of genes only. And you could see that by just assembling just those genes, basically. Something that you will not see in a paper was what happened behind the scenes And since I'm talking to a crowd of bioinformaticians, I think you will probably relate. So projects never go very easily, smoothly. Sometimes there's a problem and it doesn't work. In that case, that happened. So we ran the data, we ran the assembly. It finished without any error. But we noticed at some point that assembly was larger than expected. We expected a 2 or 3 gigabase giraffe. We obtained a 4 gigabase. Something was wrong. So we looked back at the data and we noticed that it was a little bit skewed. The mate pairs were too longer than expected, or shorter than expected actually. So the pipeline ran, but the results were off. So we went to BioStar, which is a forum for asking questions to other bioinformaticians. And then author of the tools came back to us and they said, actually do these parameters and it might work better. We did an extra cleaning, and after a few weeks of discussion and showing many data to other experts, we were able to get knowledge from the experts in that specific tool and be able to run the assembly using much finer tuning, which ended up being much more satisfactory. So this might look anecdotal, but I think for many of you who've done bioinformatics analysis might relate to this type of going back and forth to tool developers. OK, so I guess for this part of the giraffe, my message would be that if you do assembly for large genomes, it's not always necessary to have a nice finished genome, but a draft might be sufficient for the question at hand. And in that case, it was perfectly sufficient. And also be aware of tools which finish without warning. That's not always OK. Sometimes there's a little bit more quality check to be done to make sure that you have what you expect. OK, I'll mention the spruce genome assembly, which I think is relevant to Swedish interests to some extent. I didn't do so much work on it, but there's some things to say about it. So white spruce, a very large genome. So this one fits my talk quite well, I guess. 22 gigabases, but also gigabytes of data at the end. Back then, it was one of the largest sequence organisms that was planned to be assembled. And it was indeed assembled by the Abyss team in Canada, because they also have spruces, or pine trees. And in the end, the assembly was not so good in terms of quality, so 70 kb and 50 for scaffold. But it's a large genome, and so it does the job. It was remarkably annotated by an annotation pipeline which actually supported such a large genome. I think it's worth highlighting it, because normally when you run tools on larger genomes, you never know what they might do, but this one actually completed. Something notable about this process is that Abyss, even the latest version, took quite a bit of time to run this assembly. So 3 years, 3 CPU years. If you want to convert into a normal metric of time that you guess, so you divide by the number of cores. So if you have, like say, 10 cores, it would be 3 CPU months, 3 months of processing. If you have 100 cores, then it would be, let's see, a few weeks. And still quite a bit of RAM, as you can expect. So we saw this, and we say, OK, that's nice, but can we do a little bit better in terms of performance only, not in terms of quality? So we proposed a software called BiG, which actually does, oops. Oh no, it goes back to the beginning. What did I press? We proposed BiG that does part of what Abyss does in terms of assembling, but not all of it. But still, we were able to perform the unitic steps, which is, sorry, I'm pointing this way, forcing everyone to look to the left. We did the unitic steps, which right before constructing contigs. And we did it in 4 CPU days, so it's basically a few hours and not so much memory. So we are hoping that this kind of module can help you get at the start of an assembly and maybe can be extended and reduce the memory and time you needed to perform assembling of genomes in general. What's nice about Spruce is that it triggered some very clever people to look at what assemblers are doing nowadays for short reads. And in Jackman and colleagues 2017, there's a comparison of the state of the art of assemblers for short reads that can be worth looking at. This one is for human genome, but it could apply for larger genomes. On the left, there's some performance, so wall clock and peak memory for many assemblers. And on the right, there is breakpoints, so misassemblies, and a measure of NG50, so relatively N50. So you want to be on the top left. And in fact, nobody is on the top left. But it's fine also if you are, because this number of breakpoints are not too significant, so it's fine also if you are to the top. So I will not comment these plots into detail, but it's probably interesting for people who do assemblies. But just to mention the diversity of type of results you can get depending on the software you run, and also the difference in terms of resources that you can expect to wait or allocate for the assemblers. BCAM is at the very low end because it doesn't do the whole assembly process. So that's why also in terms of quality it's not so high compared to others, but it runs fast and it doesn't do many mistakes. That's kind of the same plot for the same type of software, but it highlights long-range improvements. And I think it's interesting to comment this a little. So also for human genomes, so looking at the scaffolds, still on the X there are breakpoints, and on the Y you see contiguities, so the average length of sequences. So they've taken a BS like without any type of long-range improvement, and if you improve it using BioNano or 10x, I think it's links here, BioNano here for instance, you can see that the N50 jumps from 2.5 megs to 20 megs. So a nice improvement of contiguity at the cost of not so much more mistakes being made. I won't say much more, but just to highlight that this plot exists in this paper. Those of you who want to refer to it for assembling large genomes can be useful. Oh yeah, I'll plug also my own pipeline for performing assembly because why not? It's called Minia pipeline, and you can do pretty much the same as Abyss up to the contigs. And then after that, Abyss gets a little bit better in terms of quality, but Minia is very efficient, so it can be useful to run it if you don't have sufficient resources. I probably will not have time to talk about all what's inside, and it's maybe not so interesting, but it's made of multiple small components that we have developed over the year and over the papers put together in order to have a coherent assembly pipeline. And we don't have a mascot, so I put this cat. The message for the spruce part would be very sparse, so large genomes, more complex assembly as you can expect. Still unsupported by some of the long-range improvement techniques, so 10x doesn't support larger than human, for instance. So it's still a tricky world and still a research question to assemble anything larger than human nowadays. Okay, I'll move on to long reads assembly. So long reads means either PacBio or nanopore. So pipeline for doing long reads assembly is start by error correction or not, depending on the software, then construct a large read overlap graph. So similar to what I showed for the short reads, but not exactly the same type of graph. And then do consensus refinements, so in order to correct all of those pesky sequencing errors for PacBio or nanopore. That method was previously known in the literature as overlap layout consensus. It's pretty much a synonym. Nowadays, I prefer the term string graph or overlap graph. OLC feels a bit 1990s, but it's up to you to decide. There are so many software being developed nowadays for performing long reads assembly. They work for both PacBio and nanopore, and maybe the most famous one nowadays is Canu, and there are some others. And on the right, you can see a nice assembly graph from an assembly where it seems to be that there are 2 chromosomes, right? I'll talk about the axolotl genome assembly using long reads. What's an axolotl, you might ask? If you don't, then I will still tell you. It's a Mexican salamander, for which it's almost extinct, because as you can see, they have poor eyesight. It can regenerate its limbs, so that's why it's very much studied in biology labs. So that's really cool, actually. Has a super large genome, 32 gigs. Repetitive, but not that much, 65% only. So what do we do with this genome? So there was a first prehistorical sequencing attempt from 2014. Only 19x Illumina coverage, but still costs a ton. And back then, the paper which was published in SciLifeLab said, we tried to assemble it, but we failed. There was too much resources to assemble. Okay, so that's it for, they were able to still analyze one chromosome by capturing it. But in 2016, I noticed this paper and I said, okay, let's try to do an unofficial and published assembly of the axolotl with BiG and Minia just to see what it's going to do. And we took this 19x, we cleaned it, so we ended up with 10x in the end. 10x coverage, and we ended up with a really bad assembly, just 1 kb and 50. So nothing really fantastic here. But it didn't take so much time and resources to make. I didn't publish it because really 1 kb and 50, what can you do with it? Not so much. So with more coverage, we might have been able to do a little bit more. In 2017, there was the first published assembly of the axolotl sequenced using PacBio 30x coverage. You can imagine how much that costs to sequence 10 times human genomes. And some coverage of Illumina for polishing. A whole assembler was written pretty much for this purpose, which is no small feat here. And they obtained a decent counting and 50, which they were able to improve using BioNano into a nice scaffold in 50. So that was what you get from PacBio, and using long-range improvement, then 10 times improvement in terms of size of assembly. On the right, I'm just showing a figure from that paper where they highlight the use of long reads to just span long repetitions. So some repetitions longer than 10 kb can be spanned like long reads, which makes total sense, but it's nice to mention. So what's next? In 2018 and beyond, if you wanted to assemble a large genome using long reads, then I'm guessing there's going to be many more improved assembly techniques which will come out. 2 of them have been appearing in the last month in terms of preprints and GitHub repository. One of them is WT-DBG2. That's a mouthful, which uses a notion of fuzzy debugging graph, which I don't understand very well yet. And it can assemble an axolotl using reasonable resources. Another one is RA, which is an assembler which follows this pattern on the right here. So it's a combination of multiple modules that some of you have heard of. Minimap2, a very fast mapper. Raycon, a polishing assembly tool which has been released a long time ago, and a new layout stage, so graph, assembly graph refinement step. Up. So I quickly mentioned some things about sequencing advances. Many of you have probably heard this news. So PacBio acquired Illumina. Nanopore is promising the cheaper sequencing soon enough. And there's a new type of NovaSeq class sequencer which is being introduced by a Chinese company, I think. If you are familiar with all this is not news. If you are not, my sources are Twitter and a nice blog called Omics to get nice information about new sequencing technologies. I'll also plug this technique, which was published this year, which I really like. It's called trio binning, which consists of, you want to do an assembly of a diploid organism, and you kind of want to resolve both haplotypes. A way to do this is to sequence both parents as well as the child. And by doing some bioinform magic, which consists of binning reads based on the reads of the parents, so they bin the children reads based on parents' reads, they can reconstruct both haplotypes separately. And it was established for a type of cow because they were able to cross 2 different, I don't think it's species, but 2 different parents which are far enough genetically that it's easy to separate the haplotypes. But it's also possible to do it for human genomes, and they report quality of haplotypes similar to 10x, which is kind of nice. So for instance, if you have a complex genome assembly to be done, which is diploid or even more, I think this would be the way to go. So in advance, prepare the fact that you need to sequence the parents of the organism. Yeah?
You mentioned even higher than 2 ploidy?
They don't advertise it for higher than 2. But I don't see why it wouldn't work for at least starting to separate some of the haplotypes. And probably you want to ask this question for plants and for other type of organisms. And this is still pretty much research, but just food for thought. I'll quickly plug something about assembly debugging. So once you have an assembly, this is not the end of the game. You typically can investigate the contigs, but there also exists an assembly graph reported by the assembler. It's one of the files that you might not want to look at, but it contains useful information. Like for instance, it tells you how contigs are connected. For instance, this contig 3 here looks like a repetition, which can be preceded by either contig 1 or contig 2, and followed by either contig 4 and contig 5. It's kind of useful information, and I would say that this is an informative contig graph. However, in some cases, especially in long reads, the contig graph looks a little bit like this. So you've got 3 contigs and no connection between them. When you see that, there is essentially 2 hypotheses. One is there are coverage gaps in a sequencing such that I cannot connect contig 1 to anything else, or the assembler decided to not report any connection. And it's tempting to go to hypothesis 1 because you say, well, the assembler, why didn't it report any connection? And we looked into this and we dug into this. And in the end, we proposed a few methods to actually debug assemblies. So this is a work of Pierre, who is actually in the middle of the audience right now. And we've looked at fragmented bacterial assemblies because we have to start somewhere. So bacteria are small. And sometimes they are fragmented by the assembler. And they can be debugged. So what I mean by that is imagine you have a fragmented canoe assembly, which consists of 3 contigs. It's possible to project those contigs into the original uncleaned overlap graph of the reads, and you obtain a picture like this, where the graph is the overlap graph corresponding to all the reads, and each color corresponds to a contig. And now it's clear to a little bit see what happens. So the fragmentation between the red contig and the purple contig shows that those two could be connected in practice. So we worked a little bit more to try to extend this direction, and we analyzed 45 fragmented bacterial assembly from the nice NTCT and CTC 3000 project from the UK. They sequenced 3,000 bacteria using PacBio and assembled all of them automatically. Many of them, it went fine, but for another chunk of them, I think it was 200 or even more of that, it's fragmented. So we took a few of those fragmented ones, and we did that kind of processing here when each horizontal line corresponds to a contig, and we recover some of the hidden links between contigs that were not reported by the assembly graph, and see how we can connect them. So you can see that for links which have a small number of reads between them, likely to be adjacent, and the one which have a high number of reads, more likely to go through repetition, so less likely to be real. So using this method, we were able to recover on average 35% of the contig adjacencies which were not reported by the assembler, and we hope to go a little bit higher than this. So essentially, this is finishing between quotes for free. I should have put finishing between quotes as well, because it doesn't do the finishing automatically, but it gives you some clues to what happened in terms of the assembly process. And we are looking for collaborations on PacBio sequencing to try to apply this method to real cases.
Not only PacBio.
And nanopore as well, yes. I'll also mention another event which happened last month. It was a small hackathon organized by Vancouver, where anyone could participate around the world, and we were a team of people who tried to take assembly tools and make them interoperable. So you take a module from one assembler, when you plug it into a module of another assembler, and do this with different type of assembler, and so we were able to combine spades and abyss into spabis. And VCAM and abyss gave results similar to abyss, and we did the most crazy one, which is 3 different assembler parts. GFA view is not so known, but it's from Hengli. And we obtained different assemblies, and we could in fact perform this interoperability because we used standard formats such as a GFA format, which is a representation of assembly graphs that assemblers typically output as some take as input. So far this is just a more or less a fun or experimental way, but we think it can be taken to an actual next level where we could actually design a good assembler by mixing components of the best ones available to take some hybrid assemblies and make them possible. Like, for instance, take a good assembler for short reads, a good one for long reads, and combine using this type of technology. OK, this is my conclusion part. So it's going to be a short conclusion. Assembly is still unsolved, still a research problem. But of course, long reads have greatly improved the situation. For metagenomics, it's still unclear, but I will note the tweet from yesterday where Sam Nichols and colleagues released one of the first data set of high coverage nanopore sequencing of a metagenome data set. So now it exists, and now tools can start to be developed on long reads metagenomics. So we'll see what happens in the near future. OK, and I'm making a group in sequence bioinformatics at Institut Pasteur next year, and we'll be hiring. And that's my last slide. So these are some acknowledgements. Thank you for listening, and thanks again for the invitation. And I'll take any questions.
OK, thank you very much, Ryan. Questions? Anybody have an idea? Yeah?
Which unsequenced genome terrifies you?
That's a good one. I think all the hexaploids and plants in general are very tricky, and the researchers which published wheat, for instance, this year did a really good job at doing this. So maybe the next frontier would be highly hexaploid eukaryotes, or even more ploidy. And I wouldn't know how to assemble, for instance, the metagenome that was released yesterday in long reads. So that would have been two very challenging instances for assembly. But really, if you look close enough, there are always something to be nitpicked on each, even the small instances. You can always try to improve something, depending on some cases. That's a short answer.
Other questions? Yeah?
Yeah.
So when I wanted to ask, when an assembler throws away haplotypes,
Yeah.
throws away SNPs,
Yeah.
and maybe there, as I understand it, there's a risk that haplotypes can actually end up being combined into an assembly.
Yeah.
That's actually not an assembly of one haplotype, but a combination
Yeah.
of several haplotypes, and you don't know which one belongs to which.
Yeah.
So isn't that a problem for the biological interpretation?
Yeah.
Of like adaptations and
Yeah.
genes that
So
are in linkage disequilibrium for a reason?
that's a problem. That's why recent techniques for assembly have focused on resolving haplotypes. So if you sequence using PacBio with Falcon assembler, we try to resolve haplotypes as much as possible. 10x with Supernova tries to resolve haplotypes as much as possible. But here, I'm just a messenger. I'm reporting that assemblers do it. And then biologists should know that, I mean, the human reference sequence doesn't correspond to an individual. It's a mix of multiple individuals. Like GRCH38 is a mix of people. But then you get other human assemblies for a single person, like Craig Venter, Watson, Clive Brown. But in general, assembly should be considered as a, well, if you do a bulk of cells, it's never one cell that you assemble. It's variations in between everything that you've sequenced together. Yeah. So there are ways to improve, but people should just be aware that this problem exists.
Other questions? Yes, I have one. So what about pangenomes? Using pangenomes as reference. What do you think of that type of approach?
Yeah, it's the next big thing for bioinformaticians. I think many people have been talking about it. As far as assembly is concerned, we are not assembling pangenomes. We are assembling genomes individually when possible, and then combining them is still a research question. I think the thesis of Eric Garrison is a very nice introduction to pangenome representation. So the field is not yet quite there, but people are working on how to best represent a graph of sequences. And VG would be a good way to start for the lack of better tools for now. Yeah.
Thank you very much for the talk.
Thanks.
So what do you think? Can alignment and variant calling be replaced completely by genome assembly, or is it like genome assembly will never outperform alignment? Like there are two ways of variant detection.
Yeah.
You can do genome assembly, you can do alignment and variant calling. Like how do you see which is more preferable way for structural variants, for example?
Yeah, that's a complex topic, and I'm not the best specialist in structural variants, but my vision of it is if you want to assemble insertions and complex variants, which are a mix of variants together in a single locus, an assembly-based method would perform better. If it's a simple deletion where in a region where nothing happens in the genome, then you can always call it by alignment, and it should be working fine. Alignment will never die, because even if you assemble a whole genome, you still want to align it to some other genome. So whole genome alignment has been predicted by other people to still be a booming field in the future. So that part is here to stay. Yeah.
So one final question, and then we can continue the discussion outside. There will be a coffee and some smoothies and stuff. Oh, yeah, go ahead.
Do you have tips for assembling the genome of a parasite, of an endoparasite, where you don't have the reference genome for either the parasite or the host?
So you don't care about the host, and you just want the parasite. Depending on its concentration.
It's low concentration.
Oh, that's bad. So you can try to filter out the host and keep, consider the host as a big parasite and filter out everything that is highly covered. Taxonomic classification of either the quantics or the reads would be a natural lead. GC content and small k-mers content and all the binning process that you do in metagenomes could be considered for genome host parasite setting. There's many possible leads and interesting project. Yeah.
Okay, thanks again, Ryan.
Thanks.